We sample the ecology using 3 different techniques. We sample predominantly the infauna with a Hammond grab, and that takes a scoop of the seafloor. And then we sample the epifauna with a beam trawl, which scrapes the top of the seafloor. And then we also take seabed images, and this puts the smaller Hammond grab samples into a wider context and also lets us see how those components of the marine community are arranged on the seafloor. Once the sediment sample has been collected with the grab is retrieved and brought onto the ship. Here we take the sediment sample out of the grab, and we take a photograph of the sample so that we can look back at the sample later on and see what the sediments were like and see if we can identify any obvious fauna. We then take a subsample of the sample, which we analyse for the sediment composition, and then we sieve the sample to remove any of the fine material, and the rest is then fixed with chemicals and taken back to the laboratory where it's analysed. Once the trawl sample arrives on the vessel, first we take a photograph of the trawl sample and we measure the volume of the whole catch, and then we sort the catch into different species and take photographs of them and identify them to species level where possible. With some of the smaller animals where it's quite difficult to identify them without a microscope, they're preserved and taken back to the lab for verification. So once the grabs come back to the lab, first of all the chemicals are washed off, and then the animals are separated from any remaining sediment using a microscope. And once all the animals have been picked out of the sample, they're separated into the 5 major phyla, so Annelida, Crustacea, Mollusca, Echinodermata, and anything else. It's put into another pot. And then each of these pots is identified in turn, so that every organism in there is identified and counted. And then once they've been counted, they're also biomassed. And then this data all goes into a database here and is analysed later.